innate immunity is our ability to protect ourselves from infection without any change in our body. So our immunity is divided into innate immunity, so what we can do right away, pathogen, virus goes in our body, we can immediately protect ourselves from it, that's innate immunity. Adaptive immunity is when we have to change, so we have to start making antibodies or start making reactive T cells. So innate immunity is really the first line of defense. So intracellular innate immunity is the ability of a single cell in your body on its own to protect itself from infection. And that's a very fashionable and active area of research in the last 10 to 15 years. What is innate immunity? Well, it really depends in the first place on detection of an incoming pathogen. So for example, a retrovirus. So a retrovirus goes inside a cell, and the first thing it has to do is to turn its RNA genome into DNA. Then it has to go inside the nucleus and integrate into the host chromatin. And it has to do that without the cell detecting it, without the cell knowing that it's there and activating an innate immune response. Because if the cell does detect it, then it is very capable of shutting it down. So all viruses that infect any kind of cell have to either antagonize and directly suppress or avoid the intracellular innate immune system. So for retroviruses, their biggest problem is that they turn RNA into DNA. So that's a pretty weird thing to be doing because cells are turning DNA into RNA. So a cell should be able to immediately detect the fact that RNA is being turned to DNA. The innate immune system's goal is to firstly detect viral infection by detecting something funny going on, like reverse transcription of RNA to DNA, and then to act upon that. So what a cell will do is the detection is through molecules called pattern recognition receptors, and they can, for example, detect strange-looking chemicals or strange-looking proteins that are different to what is in the cell, and they will activate a signaling cascade if they see them. There are pattern recognition receptors called DNA sensors, which detect DNA in the cytoplasm of cells, because there isn't usually any DNA in the cytoplasm. So if a retrovirus shows up and starts making DNA in the cytoplasm, typically that will activate innate immune sensing unless the virus is able to shut that process down. So if you activate innate immune sensing, then that leads to a transcriptional response. So the cell will start making proteins that it wasn't making before, and it'll start making more of proteins that it was making before that are involved in the innate immune detection. The cell will also start making proteins called inflammatory cytokines, particularly type 1 interferons. Those type 1 interferons come out of the cell, they travel off to nearby uninfected cells that haven't seen the virus yet and inform them that the virus is coming. So those cells will then activate their antiviral program, start making these antiviral proteins, and therefore there will be a ring of uninfectable cells which are referred to as being in an antiviral state around this cell that has detected the virus. And that is our first line of defense against viruses. So as I say, if a virus like HIV wants to infect some human cells, it has to get by that. So in 2016 we've discovered that one of the key things HIV does to avoid activating innate sensing is to keep its DNA synthesis very tightly inside its capsid. So the capsid is a protein shell inside the virion, and the DNA synthesis goes on inside that capsid. We've shown that the capsid is made of hexamers that are actually electrostatic channels that can suck in the nucleotides that fuel the process of DNA synthesis, which is all very indicative of the fact that it's going on inside, and that's because if it comes out, then the viral DNA is detected by the host and viral infection is shut down. The innate immune system is also very linked to the process of DNA damage. So people will say that there is not any innate immune detection of DNA in the nucleus because the nucleus is full of DNA. But on the other hand, the nucleus is very good at telling if the DNA is being broken by a virus or some other process, and that activates a DNA damage response, and that also can activate interferon and an antiviral state. So the whole process of regulating DNA damage and regulating the innate immune system are linked. And I think one of the most exciting areas at the moment is understanding how the innate immune system is linked to cancer. So recent studies in 2015, 2016 have shown that when a cancer cell is dividing in an uncontrolled way, DNA is leaking everywhere and DNA sensors are activated, and that leads to an innate immune response that can shut down a cancer in early stages. And it seems that it's a very important process of cancer formation that the DNA sensing pathways are switched off. So HIV is an excellent tool to study DNA sensing because, as I say, it makes DNA in the cytoplasm, a place where there usually isn't any DNA sensing. And we think that HIV has evolved the ability to avoid it by doing DNA synthesis in the capsid, but also by shutting down certain DNA sensing pathways. And there's evidence emerging that proteins, specific viral proteins, can have a role in shutting off the DNA sensing responses. So innate immunity is all about cells protecting themselves from things going wrong, and about triggering consequences that then either lead to secretion of cytokines and perhaps cell death as well, which will mean that that infected cell will die and disappear. So we study innate immunity by taking some cells that have innate immune pathways intact. I think one of the things we've realized is that a lot of the cell lines that we have in the lab, the innate immune pathways are broken. Some of them are broken and some are not broken. The DNA sensing pathways particularly are typically broken, and I think that that's because many of our cell lines actually derive from tumors. So the process of forming a tumor has switched off DNA sensing, and therefore when the cells get to our lab, there are no effective DNA sensing pathways. So the first thing to work on it is to make sure DNA sensing pathways are intact. And there are some cell lines that appear to have DNA sensing intact. Then you can, our favorite approach is a genetic approach. So we can, for example, take a virus and activate DNA sensing and then see if the virus can switch it off. We can then change the virus by making mutants and see if we can kill the virus's ability to switch off DNA sensing. And that's a very powerful approach to work out how viruses switch off DNA sensing. And I think certainly all DNA viruses that have been studied are quite clearly able to switch off DNA sensing. And the hope is that by studying how those proteins work, we understand the process of DNA sensing. HIV, for example, because it travels light with only 9 genes, I think its MO is really to try and avoid activating DNA sensing in the first place. One really interesting observation that's been made, I think, in around 2012, 2013, was that there is a protein called a nuclease called TREX, which actually degrades DNA but acts as an HIV cofactor. So it seemed that by breaking down the HIV genome, that helped the virus infect cells. And it made no sense, because how could breaking down the viral genome help? And it turned out that this protein TREX degrades any viral DNA that leaks out of the viral capsid into the cytoplasm. And if you stop that happening, that leaking DNA will activate innate sensing. So the virus has learned to, if it does spill any DNA by accident, it's learned to have the cellular machinery clear that up without activating innate sensing. So TREX is a protein that helps the virus by degrading that extra DNA. Now, there are some patients with a syndrome called Aicardi-Goutieres syndrome that have deletion mutations in TREX, and the TREX doesn't work properly. And those individuals are very sick from birth because they make interferon and inflammatory cytokines right from the beginning. And it's been shown that that's because the retroviruses in their body are activating those DNA sensing pathways in the absence of TREX. So it turns out that TREX's role is to switch off or suppress DNA sensing caused by the endogenous retroviruses that exist in our genome. HIV has latched onto that and can infect human cells by using TREX and that pathway to clear up the mess it makes when it infects cells. So understanding innate immunity has tremendous potential to help us treat viral infection, I think. So one of the approaches we're taking is to try and understand how the virus antagonizes innate immunity or avoids innate immunity, and to design drugs to stop that happening. So for example, we've shown that HIV-1 has to recruit particular proteins in the cell in order to manage its process of DNA synthesis. And if we stop that happening, then the viral DNA is detected by the cell. We don't understand why in that case the Trex doesn't clear up the mess, but it doesn't seem to. That activates an innate immune response, and the virus is suppressed by innate immunity. So drugs that can do that can interfere with these cofactor interactions, will have an antiviral effect by causing an innate immune response against the virus. And because that innate immune response is extremely powerful, the virus can't escape by mutating. So if the drug directly binds the virus, like most antivirals do, the virus can mutate and escape. But in this case, the drug can target the host factor, stop the host factor being used by the virus to avoid innate sensing. Innate sensing comes back online and the virus is suppressed. So the drug almost turns the situation around, so the virus is like a non-human virus anymore. It can't use the cofactor and therefore it gets suppressed by innate immunity. So I think that's one of the great possibilities in innate immunity, is going to be to learn how to use innate immunity to turn it back on against the thing that we don't like, such as cancer or viral infection or bacterial infection or anything else. The links again with cancer are very strong. So as I say, we think that very rapidly dividing cancer cells leak DNA into the cytoplasm and activate DNA sensing, and that can cause the tumor to be attacked by the innate and the adaptive immune system. So understanding why tumors or how tumors stop doing that and make them do it again could lead to a completely different type of therapy against tumors, where you're really switching the innate and adaptive immune systems back onto the tumor. And it's all going to be about understanding how innate immunity works, and then understanding how innate immunity is switched off during viral infection and during tumorigenesis. And then finally, I think the other great burgeoning field in innate immunity is the field of vaccination. So vaccination only works if you provide an antigen, which is derived from the pathogen that you are vaccinating against, and what's called an adjuvant. And it's been realized in the last 20 or 30 years that what adjuvants are actually doing is activating an innate immune response. So we knew about adjuvants before we knew how they worked. Now that we know how they work, we can improve the adjuvants, and we can make adjuvants that do activate innate immunity in different ways and see which different ways is the best way to develop a particular vaccine. So I think the study of innate immunity is really going to help us understand how to do lots of things that we couldn't do before, that are related to switching innate immunity back on to do the thing we want it to do: kill a tumor, kill a viral infection, vaccinate you.